Welcome to Science with Dr. T. This week, we'll be learning about protein structure. There are four levels of protein structure. Primary structure is simply the amino acid chain, the order of the amino acids linked together by peptide bonds. Secondary structure forms when hydrogen bonds are formed between atoms in the peptide bonds of the chain. Common secondary structures are alpha helices and beta pleated sheets. After secondary structures have formed, then the protein folds into its tertiary structure through interactions in the amino acid side chains, including hydrogen bonding, ionic bonding, and hydrophobic interactions. The tertiary structure is the functional form of most proteins. Some proteins have quaternary structure, which is when two or more tertiary structures, also called subunits, interact. In general, the same types of interactions that contribute to tertiary structure also hold the subunits together to give quaternary structure. When there are two tertiary structures, the quaternary structure is dimeric. When there's three, trimeric, and so on. Now, let's put these concepts into practice. A researcher is studying a monomeric protein that loses its function when exposed to high temperatures. Using advanced imaging techniques, they observe that hydrogen bonds between amino acid side chains are breaking, though the amino acids remain connected in their original sequence. When the temperature returns to normal, these bonds reform and the protein regains its function. Which level of protein structure was most likely disrupted by the heat? Okay, let's highlight some keywords that will help us make a decision. So monomeric lets us know that there is only one protein unit. So this protein does not have quaternary structure. So we can cross out D. Hydrogen bonds between amino acid side chains are breaking. Lets us know that this is not disrupting secondary structure because secondary structure is hydrogen bonding between atoms in the peptide bond backbone. So we can cross out B. Amino acids remain connected in their original sequence, lets us know that the primary structure is not affected. So we can cross out A. This leaves us with C, tertiary structure, where the amino acid side chains interact. Okay, let's try another problem. In a genetics lab, students are analyzing a hemoglobin mutation. The DNA sequence change results in a single amino acid substitution that doesn't affect the alpha helix regions of the protein, but prevents the protein from assembling with other subunits. Which level of protein structure is directly impacted by this mutation? Again, let's highlight some keywords that will help us make a decision. Doesn't affect the alpha helix regions of the protein lets us know that secondary structure is not affected. We can cross out B. Prevents the protein from assembling with other subunits lets us know that quaternary structure, interactions between multiple tertiary subunits, is affected. So the correct answer is D, quaternary structure. I hope this has been helpful. Make sure to practice on your own with a few different scenarios until you get the hang of it. Thank you for tuning in. Remember to like our videos, subscribe to the channel, and share. Next time, we'll look at enzyme inhibition.